Assalamualaikum dear students, how are you? Hope you are fine. Today we are going to start with the chapter 8 activity 1 transmission of genetic information. How does each of the following happen? Growth, healing of injuries, replacement of dead cells or tissues. All these are due to a cellular division called mitosis. Before we start with mitosis, let's begin with types of cells in the body. Types of cells in the body. Cells in the body are classified into two types, somatic cells or body cells and sex cells or gametes. Let's mention some examples about somatic cells such as nerve cells, muscle cells, bone cells, and others. While in the sex cells we have two types, sperm in males and ovum in females. Now, let's define somatic cells and sex cells. Somatic cells or body cells are diploid cells, which means they contain two copies of each chromosome. Somatic cells in humans have 23 pairs of chromosomes or 46 chromosomes. While sex cells or gametes are haploid cells, which means they contain one copy of each chromosome. Gametes in humans have 23 chromosomes. Now, what is mitosis? Mitosis is a type of cellular division in which a cell having 2N chromosomes gives two identical daughter cells having 2N chromosomes each. It means that the cell with 2N chromosomes divides to give two daughter cells each with 2N chromosomes. Keep in mind that mitosis is always preceded by a phase called interphase, where the cell is not dividing, it's just preparing for the division. Then, interphase is followed by mitosis. In the mitosis, we have four phases: prophase, metaphase, anaphase, and telophase. And the cell cycle is the interphase and the mitosis with the four phases. So, this is what I mean by cell cycle. Now, let's recall something. We mentioned previously that chromosomes are not visible outside cell division, so they are called chromatin, while chromosomes are visible during cell division. So we said that the chromatin condenses to become chromosomes during cell division. What do we mean by condenses? It means that the chromatin coils to become chromosomes where it is visible during cell division. Now let's distinguish between chromatin, chromatid, and the chromosome. Let's start with the chromatin. Chromatin material presents in the nucleus of the cell. It condenses into chromosomes when a cell divides. While chromosome, they are filaments contained in the nucleus of cells. They are condensed chromatin. And the chromatid, as we mentioned before, is one of two distinct units that makes up a chromosome. So, as a conclusion, the chromatin is a chromosome but in a filament structure, while chromosome is made up of two chromatids that are joined by centromere. Let's now start with the first phase of cell cycle. Let's begin with interphase. During interphase, we said that the cell is preparing to divide. If you look at the cell, what do you see? The nuclear membrane is seen. The centrosome is seen. What do you mean by centrosome? It is a cellular organelle specific to animal cell and it is necessary for cell division. The chromatin inside the nucleus is also seen. We said that interphase is followed by mitosis. Are you ready to start with mitosis? Here we go. The first phase in mitosis is prophase. What are the events that take place in prophase? If you look at the cell, what do you see? What happens during prophase? Very good. So the chromatin condenses into chromosomes, each having two sister chromatids joined by a centromere. Second, the centrosome duplicates and transfers into asters. The nuclear membrane disappears, and a spindle of fibers or a chromatic spindle forms. The second phase is metaphase. Now look at the cell. What happens? What is the event that takes place? Very good. So, the chromosomes with two sister chromatids move to the center of the cell and line up along the equator, forming equatorial plate. And this is the equatorial plate or the equator of the cell. Now the third phase in mitosis is anaphase. Now look at the figure. What happens inside the cell? Think a little. Excellent, my dears. The two chromatids of each chromosome separate when the centromere divides. The chromatids move toward the opposite poles of the cell. This is polar ascension. What do you mean by polar ascension? It means that the chromatids raise to the poles. Third, two identical sets of chromosomes, each chromosome with one chromatid, are obtained. And this is what we have in anaphase. Now, what about the first phase in mitosis, which is called telophase? What are the events that take place? If you look at the cell, so you can find that the chromosomes uncoil, lose their identity, and go back to the state of chromatin. It means that the chromosomes return to the state of chromatin. The spindle fibers break down and disappear. The asters become centrosome. A new nuclear membrane forms. At the end, the cytoplasm of the cell is cleaved in half, and the cell membrane grows to enclose both daughter cells that enter into interphase of a second cell cycle. Moreover, one important thing that takes place is the constriction at the equator. This constriction at the equator and moving inward called cleavage furrow. To sum up, the phases of the cell cycle, which are interphase and mitosis with the four phases prophase, metaphase, anaphase, and telophase. Let's watch this video.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of four phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
After watching the video, let's now move to the application. Let's start. The following figures show the phases of mitosis in a cell. We have four phases A, B, C, and D. Number 1. Identify each of the above phases. You have to give one event as a justification. Pause the video please and solve it on your notebook. Let's now move to the solution, my dears. Figure A is for telophase. Since there is constriction in the middle or cleavage furrow, you can give any other event that takes place in telophase. What about figure B, my dears? Excellent! It is metaphase, since the chromosomes with two chromatids move to the center of the cell and line up along the equator. What about figure C? Amazing, my dears, it's anaphase, since the chromatids move toward the opposite poles of the cell. Finally, figure D is prophase, since nuclear membrane disappears and spindle fibers are formed, or you can give any other event from the prophase. Let's now move to question number 2. Arrange these phases into a chronological order. It means that by their occurrence. So please pause the video and arrange them. So it is D, B, C, and A. So you have to put them in letters because D is for prophase, B for metaphase, C for anaphase, and A for telophase. So you have to put them according to their label in the figures. Let's move to the second application. Label A, B, and C in the following figures. So please pause the video to solve this question. You are fantastic, my dears. So A is for chromatin, B chromosome, and C chromatid. That's great. Now, my dears, study well the notes, and thanks for your attention. You will stay under Allah's care and His safety. Goodbye.